Odpoledne, dámy a pánové. Já teďko přejdu do angličtiny. I will switch into English because this afternoon session is with international speakers. It will be held in English. My name is Ondřej Hradil. I am the Deputy Project Director of Ceitec. And I will just introduce shortly the Ceitec scientific directors. They are sitting to my left, Professor Koča, who is the scientific director for life science. And Professor Vrba, who is scientific director for material sciences. And as this is a scientific panel, no more official speeches, as you heard in the morning, I would hand over the word to Professor Koča and Professor Vrba to give some introductory remarks about the science in Ceitec and moderate also the rest of the afternoon.
Okay. Good afternoon, ladies and gentlemen. Allow me to start this maybe presentation dealing with Ceitec project with a part concerning Brno University of Technology as one of the partners of the project. And especially we will speak about material sciences and technologies. So here you can see the 7 main programs which were prepared during preparation of application form for this project. 2 of them are dealing with material sciences and technologies. Number 1 is advanced nanotechnologies and microtechnologies. This program is coordinated by Professor Šikola, and the second program is advanced materials, which is coordinated by Professor Cihlář. Another 5 projects or programs will be discussed by my colleague Professor Koča. Because this program offered quite a huge amount of money for preparing new infrastructures, the main task of the project is to construct new buildings and equip them with new scientific technologies and new scientific instruments. It means we prepared the project which contains 7 core facilities and many small laboratories which are belonging to individual research programs, to tell the truth, belonging to research groups. According to material sciences and technologies, there are 3 of them. It's nanolithography and nanofabrication core facility. The second one is nanocharacterization. Both are dealing with nanostructures and preparing and testing of new nano devices. And the third one is concerning structural analysis laboratory. It belongs especially to research program 2. As it was explained, there are 6 main partners in the project. The biggest one is Masaryk University, which is sharing more than 50% of budget and similarly more than 50% of technologies, people and instruments. The second biggest partner is Brno University of Technology, which shares about, I think, 37% of the whole project. It means we are 2 main partners, and it is a reason why me and Professor Koča are guaranteeing these 7 programs as the main persons. Then there is Mendel University, University of Veterinary and Pharmaceutical Sciences, and two research institutes. One is Institute of Physics of Materials of Academy of Science, and the last but not least, Veterinary Research Institute. You can see, in my case, when I am belonging to material sciences and technologies, even though we are coordinating this part of project, we are also cooperating with our colleagues from another partners. It means, especially from Masaryk University, there is a strong team dealing with nano and microtechnologies. Then a smaller but very important team belonging to biosensing technologies from Mendel University, and of course a strong group from Institute of Physics of Materials cooperating with my people from Advanced Materials program. When we are speaking about buildings, there will be set two new buildings or two new areas of buildings in both main universities. It is a map showing you in red. Here, it's a future position of Ceitec buildings in campus of Brno University of Technology. Especially, it's a very good position for us because here are concentrated our faculties. In yellow, there are 3 new buildings under construction now, belonging to Faculty of Electrical Engineering and Communication. And here, between Faculty of Business and Management and Faculty of Chemistry will be new Ceitec. Here is the view, and you can see also the rough budget for our group, 80 million of euros. Here are 3 main buildings. This one will be equipped with clean rooms and everything concerning nano and microtechnologies. Here we'll be building with technologies and instruments for ceramic and composite materials, and this one for polymers. This part contains not only administrative offices but also computer rooms and other supporting technologies. A few words about 2 first programs. Advanced Nanotechnologies and Microtechnologies, which is coordinated by Tomáš Šikola. And we will be focused to everything dealing with nanostructures, nano devices, nanoelectronics, and nanophotonics. It means not only preparation of new nanostructures and their characterization, but also a special interest will be done to biomedical applications. And last but not least, we will be preparing new such types of structures for energetic information and communication technologies. It means it defines also our application potential in these mentioned areas. We will be, for instance, preparing new micro-electromechanical and nano-electromechanical systems for special applications, biosensors and standard types of sensors, and especially some new special electronic circuits on chip, so-called lab on chip. And of course, special interest is done for semiconductor metallic and magnetic nanostructures, including nanotubes and nanowires. As I told, there will be clean rooms in class from 100 to 100,000 with nanofabrication technologies for top-down or bottom-up methods, and of course nanocharacterization and description of surfaces and nanostructures. Here are some pictures dealing with our previous research results. This one is illustrating structures which are prepared by SPM nanolithography using local anodic oxidation for preparing new types of structures, especially here in silicon surfaces. Another possibility how to prepare similar nanostructures is here. It's focused ion beam and focused ion beam chemical vapor deposition. Here is a very strong cooperation with Tescan and FEI companies dealing with microscopy. Another example, it's especially application for medicine. It is separation of viral nucleic acids for viruses detection. And the second research program, coordinated by Professor Cihlář, is dealing with materials, especially metallic, ceramic, and composite materials, plus polymeric, new types of polymeric materials. It means our application potential will be advanced polymeric, ceramic, metallic, and composite materials for industry, energy, and medicine. We will test and prepare the materials for new thermal and mechanical parameters and properties. Plant infrastructure, structural analysis by TEM and SEM in high and low vacuum, X-ray diffraction for analysis of phase composition of inorganic and or organic materials, textures, etc. For instance, now we will be purchasing new microtomography system. Again, some examples: bioceramics, electroceramics, and materials which are compatible with human body, for instance. Some new devices for fuel cells and special applications with transparent ceramic. Here are some pictures about nanostructured coating for implants for bodies, human bodies. The last slide deals with a special group inside the materials for sensors. It's dealing with special technologies, everything dealing with systems above sensors and collecting into systems for embedded applications and robotics. So, from my side, it's everything. I would like to ask my colleague, Professor Koča, and he will tell you a few words about 5 following programs.
Thank you. Good afternoon, ladies and gentlemen. My task is extremely difficult because, having a look at my watch, I have to give my short talk in -5 minutes. Please do allow me to have some positive time. When looking at the audience, it's, I wouldn't say substantially, but it's just less people in the morning than in the afternoon, and I was wondering why it is so. And I think I know the answer. In the morning, there was a prime minister here offering money. In the afternoon, there are scientific directors here offering work. So, but still, people enough to work. I will try to offer you work in 5 scientific programs, 5 research programs that are more close to life sciences than to material sciences. The first one is structural biology. Then there is a genomics and proteomics of plant systems, molecular medicine, brain and mind research, and molecular veterinary medicine and molecular medicine. As Professor Vrba, I'm going also talk briefly about core facilities that we would like to create and run during the 5 years. It's going to be it's going to be core facility for nanobiotechnology and biointeractions, which will mainly be based on atomic force microscopy, surface plasmon resonance, and microcalorimetry for biomolecular interaction studies. It's going to be core facility for protein crystallization and single crystal diffraction that will be equipped by 1 or 2 diffractometers, and also quite good instrumentation for automated crystallization of proteins. There will be core facility for animal spectroscopy. I will talk about a little bit more later. And related cryo-electron microscopy core facility. There will be a core facility for genomics that will mainly be collected or equipped by a series of high-throughput sequencers. There will be core facility for proteomics, mainly focused or equipped by mass spectrometry instrumentation. And there will be core facility for molecular and functional imaging, where we would like to have 2 magnetic resonance instruments. One would be 3 Tesla instrument, and the other one 7 Tesla instrument. Let's go briefly through the research programs. The research program of structural biology is coordinated by Professor Sklenář. The keyword for this program is to collect as much as detailed and different level information, 3D information about molecular assemblies that are mainly composed of proteins, nucleic acids, carbohydrates, and lipids. And that's one thing, and the other thing, to relate this information with the biological function. Certainly, there is a quite strong application potential that will, within this program, that will mainly be focused on design of biologically active compounds, especially drugs. That's one direction. The other direction would be biosensing and this kind of research. Core facilities were already mentioned. So structural biology will be responsible for several core facilities you have seen in the previous picture. I like to highlight NMR spectroscopy core facility because it's going to be equipped by 950 megahertz machine. It's going to NMR machine is going to be the first one in the former, they call it Eastern Bloc, and it will become, I would say, without any doubts, the best laboratory in the former Central and Eastern European countries, and it will become also a member of this nice ESFR family within the program Instruct. Key technologies within structural biology I already mentioned. You saw everything, yes, on the previous pictures. Research program of genomics and proteomics of plant systems is coordinated by Professor Fajkus. The key point is to keep the tradition plant research has here in Brno. You see the key or the general research orientation. It's one of the keywords would be evolution. Evolution in biological systems studied on the model plant systems. And of course, there is a strong application potential of this research program that goes to, say, renewable energy resources based on plants, production of biopharmaceuticals based on plants, etc., etc. This research program will be responsible to create and run the proteomics core facility. As you see on this slide, you see selected research directions. You see that it's quite widely based program. The responsible university will be Masaryk University and Mendel Agriculture and Forestry University. You see here the words like genome, developmental biology, hormonal regulation, research on telomeres, and also on chromatin, and also you see that the last paragraph, which is metabolomical studies that will be realized on plants and also on bacterial systems. And then for Ceitec, this small paragraph is a bridge to systems biology area, which is quite weak in the country, and we believe it's going to be important. It's already important. It's going to be stronger in the Czech Republic in the future. A research program of molecular medicine is coordinated by Professor Pospíšilová. If I want to briefly summarize what this program is about, I would say it's a genome-related everything. It's a genome-related, especially genome-related illnesses, a genome-related treatment. And of course, it has, again, strong application potential. It goes to diagnosis, it goes to therapy, it goes to things related to the human health. Molecular medicine is one of the two research programs that bridges the basic research within Ceitec with medicine. So it leads us to applications in human medicine. Again, what I already mentioned, the keyword for this research program is genome and its relation to different kind of body, etc. A research program of brain and mind research is coordinated by Professor Brázdil. It's a program from the field of neurosciences. The important thing about this program is, again, it's widely based, and it starts on the one hand physically with the human brain, with samples of the human brain. On the one hand, on the other hand, it goes to things like, behavioral, cognitive level of research. So it's quite wide. I'm sure we are not on any doubts about application potential, especially these days when the population becomes older and older, and illness that was not visible 50 or 100 years ago now becomes a serious problem. This research program will be responsible to create, to set up, to run the core facility of magnetic resonance imaging, as I already mentioned. Briefly, main goals. I think I will skip the picture because of time pressure. The last but not least is the research program Molecular Veterinary Medicine, which is coordinated by Professor Hořín. This program is based in Veterinary and Pharmaceutical University and Research Institute of Veterinary Medicine. One of the keywords also in this program is genome-related things, but in veterinary medicine. I would say it follows two branches of research. One would be to follow pathogens in the food chain, and the other one to study illness in veterinary medicine. Again, consequences in the human life is quite obvious. This research program is not responsible for creating a core facility as it is. It means generally used facility for other research programs and for the region. But it will also, of course, it will also need its research infrastructure, so new laboratories will be built, but they will mainly be used by the research program itself. Some examples of research orientation. You see that you see again pathogens here. You see immunity and things like that. This picture you have seen several times because of what? If you want to do science, you need to have a good subject to study that has a future, that is competitive. Then you need buildings. And then you need what? You need people, of course. That's the key word, that's the key point. You need people. Ceitec is a young team. You see some randomly selected examples of research group from the program of structural biology. You see at the NMR group, you see the protein RNA interaction group, RNA processing research group, glycobiochemistry research group, computational chemistry research group. So you see Ceitec is from the beginning quite a young team, but we wish to take care also about very, very young generation to make sure that the future of Ceitec is a good future. And that's a proof. With this statement, I like to finalize my presentation. And I think I can smoothly continue to invite next speaker. Next speaker is Dr. Silke Schumacher. I had to do some homework because it was just last minute I realized that you will come because the director unfortunately had some problems and couldn't come. Briefly introduce you. Dr. Schumacher graduated from Paris 11 University. Yes. Good. And then she went to Bethesda to spend some time at NIH. Mm-hmm. Then she went to industry and she went through several positions in, for example, in pharma business development with Merck, then with Anadis Pharmaceuticals Europe, and then she joined EMBL. It was 2003, became head of international relations from 2005, and the director of international relations from 2010. The floor is yours. Thank you. Will take it?
Okay, so good afternoon. And first of all, I have to send warmest wishes on behalf of my director, who unfortunately was not able to be here, Ian Matai. And he would like to, and I on his behalf, congratulate you on this day, on this occasion, on this wonderful venue, on the good weather, and on the future success of Ceitec, which is off to such a good start. What I'm going to do now is I'm going to talk a little bit about my own institution, EMBL. I'm going to give you an overview of the different research activities, and at the end also talk about future possibilities for cooperation. So EMBL is an intergovernmental research organization. It was established in 1974 by originally 10 member states. Today we have 20 member states and one associate member state, Australia, which is a non-European country. You can see that most of the member states are from the Western European countries, and we are very much looking forward to extend our membership also towards the Central and Eastern European countries. So in EMBL, we are located in 5 different sites in 4 countries, so it's quite a large organization. We have more than 1,500 staff from over 70 nationalities, and the main laboratory is in Heidelberg in the the campus of EMBL with a main research laboratory and the central administration. And since last year, we also have our new advanced training center, which offers possibility to organize conferences. We share the site with our sister organization, the European Molecular Biology Organization, which is more an academy-type organization that provides fellowships for young scientists and also for courses and conferences and workshops. Then we have two what we call outstations that are located at sites of large synchrotron facilities, one in Hamburg on the site of the DESI, the German synchrotron facility, which is currently has just been upgraded to a new third-generation synchrotron facility called Petra 3, where we are in the process of building new beamlines for small-angle scattering and protein crystallography, and we'll be offering services gradually starting towards the end of this year. And the second facility, the second outstation of EMBL is in Grenoble on the side of the ESFR. Serge will be the next speaker. He will tell you all about it, where we have also a small site with about 90 staff that are collaborating very closely with the ESFR and the ILL and the Institut de Biologie Structurale, which is a French institution. We've set up a partnership for structural biology in Grenoble, which combines technology platforms from all of the partner institutions that share the site. And this is, again, a very interesting service that is being provided not only locally, but to the European scientific community. The largest site, second largest site of EMBL is located in Hingston on the campus of the Wellcome Trust Sanger Center. And this is the European Bioinformatics Institute, where currently 500 people are working. This has been the most rapidly growing site of EMBL, part of EMBL. Bioinformatics, biology is creating huge amounts of data, and the EBI has doubled in the last 5 years in terms of staff and size of compute capacity and data storage capacity. And then we have an outstation in Monterotondo in Italy for mouse biology. EMBL's missions are to carry out basic research in molecular biology. This is the main purpose of the laboratory, but we're also engaged in service provision, namely in bioinformatics and in structural biology. We have a large advanced training program. We train pre-doctoral students, postdoctoral students, and then also more advanced scientists. We engage in the development of instruments and technology, have also a very active technology transfer arm, and we are, as the only European laboratory for molecular biology, we are very much engaged in activities that foster European integration. In terms of scientific excellence, everything that we do is based on excellence, and in this ranking that shows data from over a 10-year period from 1999 to 2009, EMBL ranks as the best laboratory in molecular biology and genetics in Europe based on citation indices. We have often people that ask why EMBL, why is it different? What is this in comparison to a national research institute? And often we have to explain what the difference is or what maybe is our unique selling point. And what is really important is that we are able to hire the best people. We don't have any quota on nationalities. We can take whoever is the best in his or her field, and this helps us to establish very good scientific reputation. The fact that we combine not only research but also training activities and service provision in one institution we think provides very good synergies that help both the research activities as well as the service provision and training activities. We have a continuous staff turnover. Nobody in the institute can be employed for more than 9 years. Only 10% of the staff has longer term contracts, so we have a continuous turnover, which allows a lot of flexibility, but also allows to build a network throughout Europe and the world with scientists that have spent some time at EMBL and decide to go on to pursue their careers in their home countries or elsewhere. We have very stringent quality control through a scientific advisory committee that is appointed by our supervisory body, and we give the research groups a lot of financial and intellectual freedom. In terms of research, we cover everything from genomes to proteins, cells, and embryos, mostly using model organisms such as Drosophila or mice. But of course, a lot of the research that is carried out today in basic biology research has applications to medicine. And we have established a number of external collaborations also to enable us to bridge the gap between basic research and medicine a little bit better. In the next 5-year program, which will officially start in 2012 and will run to 2016, we see a number of areas as becoming more and more important and requiring additional support and more focus. One is imaging, where we are trying to bridge the scales between lower and higher resolution imaging techniques, where we're trying to have 4-dimensional imaging, not only 3-dimensional space, but also time, and where more and more quantitative data will be generated, which is a huge challenge. Computational biology has become almost ubiquitous. It's everywhere in all the different research departments, research groups. People use computational biology methods, and we need to build on that and continue to integrate it. Next generation sequencing has been mentioned, is of course something that has become essential, and we will also continue to expand our activities in this area, and disease models and mechanisms I mentioned. I will show you one example of a recent research project that was carried out under the leadership of Per Borg, where the human gut microbiome was investigated. There is a very large quantity of bacteria in the human gut, and this project took an approach that is called metagenomics, where you basically sequence and analyze the genetic material of the entire gut microbiome. What they found is that humans can be classified into 3 different types based on their gut microbes, and this led to the identification of a number of microbial gut markers, which can hopefully be used in the future as indicators to help deal with disease, diet, and other issues. In terms of services, EMBL provides services mainly in 2 areas. In structural biology, we have more than 3,000 users, which most of them actually are visiting ESFR, where we work together with ESFR to help provide services in structural biology, but also at the other side in Hamburg, where we have a smaller number of users. And then the second area is bioinformatics, where the services are provided not by people physically visiting one of our sites, but through the EBI's website, which receives more than 5 million hits per day at the moment. In terms of technology development, we've developed technology in many different areas over the history of the laboratory, very much driven by the need of the researchers to have better techniques in synchrotron instrumentation, software development, or imaging. These are just examples. And there's very good cross-fertilization between the the technology development and the research activities. This is now working, it's good. So this is just one example of images that were acquired with a microscope, a spin microscope that was developed at EMBL by Ernst Stelzer. And this shows you the development of a zebrafish embryo over the first 24 hours, where each little dot is one nucleus of a cell, and where you can see how the cells divide and then form shapes. And this is a three-dimensional image where you can basically over time see the development and track also individual cells, the fate of individual cells. We have a technology transfer company that is operated at arm's length of EMBL that handles all intellectual property rights for EMBL to either license it to companies or in some cases to help start up new companies. The training activities are split into intramural training, which includes the international PhD program with about 200 PhD students from 40 countries. We have about 220 postdocs and also offer a general training program for all staff members. The extramural training activities include the courses and conferences, the learning laboratory for the life sciences, which is a training activity for teachers, and then we have also more senior scholars and visitors program. I talked about the staff turnover and the fact that EMBL staff can only stay for a maximum of 9 years. We have today over 5,000 alumni that are spread across the world, about 3,000 with known addresses, and we can say that over 80% of these alumni live in EMBL member states. So this is one of the return of investments that the member states get from EMBL. ESFR projects were briefly mentioned, and EMBL is participating in 7 out of now 13 or 14 biomedical research infrastructure projects that were on the list of the European Strategic Forum on Research Infrastructures, ESFR. From the first round of projects, we are coordinating the Elixir project, which is aiming to build a new infrastructure for biological information. And in the second round of projects, we are coordinating the Eurobioimaging project for biological and medical imaging. We're also participating in many, in several others. Instruct, as been mentioned, is also a very important project for us, and we've been very closely involved in helping to move Instruct forward and to build a network of structural biology facilities throughout Europe. So now I'm almost at the end of my presentation. I would like to mention a few areas of possible collaboration between EMBL and Ceitec. In fact, we are in the process of organizing a workshop here in Brno on 21st of October to bring together EMBL scientists with scientists from Ceitec to discuss possible collaborations in a number of areas: structural biology, genomics, protein expression, light microscopy, and computational biology, as well as developmental biology. And you can see this almost fits perfectly your list of subjects of interest. So I think we will have lots of opportunities to discuss research projects in more detail and to start new collaborations. There is already an existing collaboration between Jan Ellenberg and Martin Anger, and we have actually had a number of visits from people from Ceitec, both the management and also scientists, to EMBL, and so some of these interactions have already started. We are currently in the process of discussing membership of the Czech Republic in EMBL. We had a meeting at the Ministry of Research and Science and Research on September 9th, and my director met with the Vice Minister, Ivan Willem, to discuss the possibility for Czech Republic to join EMBL, which would be highly desirable from our standpoint, and I think a lot of support. I've heard a lot of support from people here in the room and also at the meeting that was organized in Prague a few weeks ago. And there are lots of advantages for Czech Republic to join, and I think it would be very good if this would happen in the near future. And this is the end. Thank you.
Thank you very much. Even if we are under time pressure, I think we can allow one quick question, if any. If there is no question, I would like to say that about this membership of the Czech Republic, there are certainly many important people here. If you will talk to the people at Ministry of Education, tell them that the membership of the Czech Republic in the organization is very, very important, so they will just try to push them. You have seen that it's really an exciting institution. Thanks a lot. And then I have it's a great pleasure for me to be able to invite the next speaker, who is Dr. Serge Perez, Director Scientific of European Synchrotron Radiation Facility in Grenoble. Thank you. Dr. Perez graduated from University in Bordeaux, and already at that time he has shown himself to be a very strong personality, because he left this beautiful area to the United States of America when he spent 1 year in Oregon as a postdoc, and then he switched to University of Montreal in Canada for other 3 years as a postdoc. He returned to France, he returned to Grenoble, got a position at the institute which is called CERMAF, which one of the best institutes oriented to carbohydrate research worldwide. And then he spent 1 year sabbatical in the United States working in industry for Kodak company. Returned to France again, spent some time working for institution that is performing agriculture research, which is called INRA, and then he accepted the director position at the CERMAF Institute back in Grenoble. Spent 12 years on that position, and I think 3 years ago he accepted the position of Scientific Director of European Synchrotron Radiation Facility. So he spent a long time in top positions, but he is a good example. Even if you are spending time in top positions, you can do science. He has published more than 250 papers. Serge, the floor is yours.
Thank you. Yes, thank you very much, Jarek, and thank you very much for having the honor and the pleasure to be here with you. I've been that's okay. I've been following the Ceitec project, almost from this very beginning, 2004, because we had a lot of collaboration, and we are speaking about the way, how to also to improve the science and the structuration of science at the European level. So it's very pleasing to be here today and to see, this very exciting time whereby, structural biology, material science, and many disciplines are being put together. And in this cross-disciplinary endeavor, we expect, just that good science will be resulting, and also setting up, I would say, a scientific platform in Central Europe, which is also very essential for structuring the European research area. So for these two reasons, as for personal reasons, and of course, for being today in charge of the Scientific Direction of Life Science at ESFR. It's a great honor to be here. And I like to share with you what I'm calling what we can offer. And because Czech Republic is already a member of these large-scale facilities, but maybe not even known, what we can offer, as I said, in terms of providing European light for science or light for European science, whatever you want. And so in a sense, I'm not speaking only on behalf of the ESFR. I'm being the spokesperson of what we call the European Photon and Neutron Campus, which results from the synergy and between the ESFR, between the ILL, and also, as Silke mentioned, between the outstation of the EMBL and also other very, very important research laboratory. So, what are the mission of this ILL and the ESFR? And so, why do we need the European X-ray and neutron sources? And of course, these are, as we call, they are users' facilities. So our mission is to have users, and we are actually dealing with a user base of about 20,000 scientists all over Europe and also a little bit outside Europe. And our mission is to provide the best tool for the scientists to get the best science out of that. And so these large-scale facilities are based on excellence. And they are meant to provide these unique opportunities for performing cutting-edge research in the field of structure and dynamics of matter. And of course, we can, with the use of neutron, with the use of X-ray, we can cover the global aspect of matter, irrespective of the nature of this matter. This matter can be organic, can be inorganic, can be biology. We can deal with the elucidation of the structure of this matter. And so our cases have been led some time ago because the construction started, and over the years, they have been updated in a very regular manner just to maintain the high level of excellence. So in terms of major neutron sources, so this is a map which describes where we are in terms of neutron sources on the world. And so there are 12 in Europe, 6 in North America, and 5 in Asia. And the most powerful one is the so-called ILL, Institut Laoué-Langevin, which is in Grenoble. And this is the most powerful neutron source in the world for research. And there will be another one coming along, which is a European spallation source, which is meant to be constructed in Lund by the 2020. So this major source is accompanied by the X-ray source. And this one also is located in Grenoble. So Grenoble is the only place on Earth where on the same campus you have the neutron facility and you have an X-ray facility. What I said today, this morning also, Grenoble is about the same size as Brno, about 500,000 inhabitants. And so this shows that, by investing a tremendous amount of resources into this pretty small city, one can really create significant values in terms of providing new ideas, in terms of providing new data, and of course in terms of translating that towards the society and towards also maybe industry. So in terms of X-ray source, the ESFR was the first generation of hardware source. And of course, this is not the only one in this category. In the first league, we have not only ESFR, but then we have APS in the USA. We have also Spring 8 in Japan. And as Silke mentioned, now there is the upgrade of this Synchrotron facility in Hamburg, Petra 3. And so this, along with ESFR, this will form the group, the four big Synchrotron facilities. However, there have been a significant number of construction of other European sources, X-ray sources in Europe. There is, for example, Diamond in the UK, Soleil in France, there will be Alba in Spain. And these sources operate at the middle range energy. So in a sense, they are not competitive with respect to this high 6 GeV machine. They are complementary. And this is how the construction of this European X-ray facility has to be understood. So there is no competition, there are complementarity. Of course, sometimes people from politics want to put us in terms of competition where they should put us in terms of complementarity. However, now we are producing data, we are producing knowledge, and we want to compare what are the yield between these centers. So this is a number of publications which result every year. And so you see, if we take APS, I mean, there are about 1,000 publications which are published every year. If you take DESI, because DESI is a growing facility, that's 500. Diamond is about 300. Spring 8, which has a higher energy than ESRF, is producing about 400, 500 publications. And in our case, we are publishing 1,000, almost 800 publications. So this results from about 1,500 experiments. So these 1,500 experiments are, at the beginning, that generate 1,800 publications. So which is, of course, how we quantify, how we compete, in a sense, or we compare ourselves in terms of efficiency with other synchrotron facility. And so this is, this is because we are committed to excellence, both at ESRF and ILL, in terms of scientific program, in terms of the staff which we are recruiting, and like Silke was mentioning from EMBL, we have a fairly strong turnover also of scientists. We hire very good scientists. They can go for PhD training, they can go for postdoctoral training, they can stay for a while, but not all of our scientists are going to be staying on the site. They have to go to other places. And this is, of course, the key words for the excellence and the overall turnover at the facility. We have a strong competition for beam time. Beam time is allocated based on excellence. Our user base is about 20,000 scientists. And every year we receive about 6,000 scientists on our campus. So not only we have to provide neutron and X-ray, we have to provide good food, we have to provide lodging, because this is essential. I mean, if you have to work, you can come and we will pay for it if your beam time has been accepted. You can come on the site, you can share with your team. We can pay up to 3-4 people. They will be sleeping, they will be working on the X-ray beam, they will be nicely fed, and they will be the most efficient. So this is also a key to the success, and that's maybe something that you have to also integrate in the management of the attraction of Ceitec. And of course, we also have under strong, constant, I would say, evaluation from the financial aspect, from the administration aspect, and also for the scientific outcome and how we foresee the future at these places. Few words about ILL. So this is a very important budget. This is the annual budget of about 80 to 90 million euro. After Fukushima, maybe they're going to be needing more money because, everyone is concerned with the safety of neutron facility. ILL received about 1,300 proposals per year, out of which only half are accepted, and also host about 2,000 visits. We are sharing the same, I would say, the same hotel, the same restaurant, the same cafeteria. In 2008, from the experiment, they had about 600 refereed publications, out of which 120 in highly impact publication. Again, as we said for ESRF, ILL is comparing itself with other neutron sources. And from this slide here, you show these are the outcome in terms of number of publications. And this number of publications, which is high, again, ILL is one of the most productive places in terms of science on the world, in the field of neutron. And out of this publication, they have a significant amount, maybe 120, which appear in a highly ranked journal. By highly ranked journal, I mentioned Nature, Science, Cell, PRL, JAX, and so forth, macromolecule if you do polymer science. How about ESRF? ESRF has a slightly larger budget. We are in the 80 millions to 100 millions per year because we are running an upgrade program. We receive about 2,000 proposals every year, out of which 900 are accepted. And these 900 proposals are giving rise to this 1,500 experiment, which I was describing to you. And in 2008 and 10, we identified 1,800 publications, out of which 200 high-impact publications, and especially true in the case of structural biology, because our users publish, and we have almost every week one paper which is published either in Nature or Science, which comes from experiments which has been run at ESFR. And structural biology is a key component. So structural governance, so this is maybe a little bit of information for you because the Czech Republic is a member of the ILL in the context of a consortium which has been signed between Czech Republic, Austria, Hungary, and Slovakia. So in a sense, one can apply, the scientists can apply, the beam time is allocated, everything will be paid in terms of the travel, in terms of the housing, hosting of the students and the researcher. And as for ESFR, Czech also is a member is an associate member of the ESFR at about 1.05%. And this is in the context of what we call central sync, which means synchrotron in Central Europe, which is between Czech Republic, Hungary, and Slovakia. So, what are the type of science that we can offer that our users are running? Of course, as I said to you, the complementarity between neutron and X-ray is key for investigating matters and materials. And of course, as we all know, these matters are made of atoms. And of course, if you are capable of characterizing the location of the electron and the nuclei, you have everything for the characterization of the structure and eventually, in certain cases, of the dynamics of your system. So we are providing these tools for understanding materials at the atomic length and, of course, time scales also, and in order to answer fundamental and applied questions on the matter which is surrounding us. And this is a slide which shows that over the ages, the technological progress have been very, almost often, or always linked to progress in understanding material. So this is a little bit of history. I mean, we go from Stone Age, we go from Bronze Age, we go from Steel Age. And every time we have been understanding and the mastering of the material, this has been translated into a significant progress. Nowadays, we have been, maybe we are still in the oil age, but we are also facing the silicon age, and we have to be able to be the best in terms of characterizing and using these different types of material. So this is what we are providing. And of course, if we can provide that, then you have all the ingredients for designing materials based on knowledge. So the design material can cover many aspects. It can cover the aspect of fundamental chemistry, whereby you can also assess some key processes arriving in catalysis. And some Japanese industrial Toyota came to our beamline and developed during 1 year a research program, at the end of which they took one patent, they published 3 or 4 publications, and they understood the basic of the catalytic process. They returned to Japan, where they built their own beamline now to continue. So they are key to understanding the catalysis. Of course, this can be key also to understand how our cell, is moving on a nano-pattern. This is nano-patterning substrate, and understanding how the cell is moving can be also key to many applications, fundamental understanding and application. Treating osteoporosis, characterizing osteoporosis is also important. This is as important as characterizing the material for building this nice plane and so forth. And of course, if you can do so, then you have this famous way of going on characterization of a sample and characterization of the structure. And of course, you want to link the structure to the property, and you want to link this property to performances, and of course, to processing. And by having this information, you are capable of providing, designing, improving new materials. Now, what we want to do that in real time, in situ, and in operando, because we think that if we are capable of facing time scale which are short enough just to provide the user with almost an immediate answer with respect to the experiment which is being conducted, this will be also tremendous advancement in terms of designing new materials. And of course, we can investigate the matter and material. So the fields of application are manifold. I just shown here in the field of solid-state physics, nuclear and particle physics, chemistry, medicine, and life sciences. So these are the field of application which can be covered with, of course, many applications. And if I talk about medicine, of course, I can talk about pharmaceutical molecules. I can talk about new therapy protocols. If I talk about life sciences, I can talk about new colloids which are capable of delivering drugs and so forth. And so this is what we can do, and we have also vision for the future. Vision for the future, this can be seen only in the context of how we put the science in the context of the 21st century. We think we are reaching a stage whereby these large-scale facilities are no longer the playground of physics. They have to be used by chemists. They have to be used by people in medicine and so forth. And so we have to be capable of showing the society that we are addressing matters which correspond to the today's challenges. What are the today's challenges? We are facing environmental and climate change mitigation. We also have to face the energy and food supplies for the 8 billion people. We have also to provide affordable health and new drugs to combat disease, not only to the rich Western world. We have also to be capable of providing sustainable transport and space where to live. And of course, information sharing and archiving is also essential. So this is how we foresee the future of our beamline, of our operation. And both at ILL and ESFR, we have been setting up research program, of course, in collaboration with the scientific community, which deals with the field of structural biology, which can be, of course, having input in material sciences and energy research and so forth, and health. So these are the key areas of research which we are addressing: science at extreme condition, chemical changing and material processing, nanoscience and nanotechnology, whereby we are building very new, very long beamline. One of the beamline will be about 200 meters long, capable of performing nanoanalysis and nanoimaging, X-ray imaging, both for material sciences but also for medical application. Of course, time-resolved experiments, which are so important if you want to address issues whereby your material is being investigated in operando, magnetism and electronics, and nuclear and particle physics. And we are also carry on studying in the field of radiotherapy. We are using the synchrotron beam as a way to treat some brain tumors. And we have two research programs going on in this field. One is called the stereotactic synchrotron radiation therapy, whereby the interaction between IZ elements like iodine, gadolinium, and the interaction of this X-ray beam are going to be capable of destroying in a very limited place the tumor, and this is aimed at treating brain tumors. And we are developing this program in collaboration with the Institute of Neuroscience in Grenoble and also the local hospital, because we need the complementarity between medical doctors, between neuroscientists, between people doing oncology. And we are ready now to have our first patient. So we are going to enter in some sort of a shutdown because of the upgrade, but we should be capable of having our first patient being hosted and treated within the phase 1 program to be treated. We have a second program which also is called microbeam radiation therapy, whereby we are addressing the possibility of delivering a very high dose of radiation, which are being delivered in a very spatially fractionated mode. And this is also aimed at curing brain tumor, which cannot be cured using normal therapy. So these are very important things. So we have animal house, we do histology, molecular biology, and so forth within this. And we can, of course, host your mouse and so forth. And I can tell you this is the most complicated research program to defend, because if you talk to people doing physics, they don't mind being sitting nearby the most powerful nuclear reactor. But as soon as I see a rat, they don't understand. And it's very, very difficult to deal with these two different worlds. And also we are doing something different. We are doing paleontology and cultural heritage, and whereby we are using a new technology which are no longer based on the contrast between intensity, but phase contrast. So this can allow, for setting up a complete tomography at a submicron level. Oh yeah, I know she's not happy, I will tell you why. At the microradiation level, a unique object. So this is a unique object which is Sediba, as 2 million years old, we can construct the three-dimensional structure of these unique things for humanity, and we are creating a virtual museum without destroying the sample. So these are some of the few things which we've been doing in paleontology. And so we also addresses the issue of cultural heritage, and there are some examples which have been addressed. So this is a Van Gogh painting which has been fading away along the year. So what are the chemical reactions which occur and so forth? And of course, the Mona Lisa is not happy because we have been using not our X-ray beam, but also a specially designed computer program to understand the famous sfumato that Leonardo da Vinci was using just to give to Mona Lisa this very unique picture. We have a special edition which I can share with you about that. And this is very important because this is public outreach. With this example, we can go to the public. We can discuss to the youngster. We can go to the citizen and say, we are doing a very good science in terms of physics, in terms of chemistry, but you see, we are also doing science in terms of cultural heritage, and this is what the person understands. So in doing so, we get a support of the societal support, which is so essential. So, and this is now what we can offer in terms of partnership and collaboration. Silke has already addressed the issue of collaboration which you have put forward in terms of the structural biology. So this is our campus, and the campus where we have the ring where we have EMBL is located here. This is the neutron scattering, and I would say like the region here. In Moravia, the local region is investing a lot, and we had the support of 80 million euros just to create a new infrastructure, just for improving the quality of reception and the science which is there. And this is the aim for scientific partnerships. And this is what I would like to finish my presentation with, about how we see the science. We have set up this partnership in structural biology. So this is a common history between EMBL, ESFR, ILL, and IBS, and of course there are new members, and whereby we can propose to our members, to carry on experiments, and by having access to neutron scattering to all these platforms. We are dealing with more than 15 platforms that cover the complementarity between neutron and X-ray. They go all the way between biophysics, but we can also have an automated system for soluble expression and crystallization of protein and so forth. And so this is a key center which is considered as a magnificent example, which is being copied by our colleagues in England, by our colleague in Germany, and whereby, as a member, you can send students to this place in the context of the membership which exists. And of course, this allows to cover many aspects of structural biology, and also we're going to have access to a 950 megahertz machine and electron microscopy. So not only we are dealing with the best of structural biology, but this allows to enter in system biology in the next coming year. We are doing something slightly different. Now we are promoting the concept of research hotel, especially in the field of soft condensed matter. If you do polymer science, you cannot send your polymer sample, by DHL. So you have to be able to come to the site, have access to a laboratory, prepare your sample, then characterize this sample by using X-ray or neutron. And once the structural characterization is performed, you can characterize the property or the function of these. And then you enter again in this cycle of engineering. In order to do that, you have to be capable of being located on site. And we are building the so-called science building, whereby in the concept of this research hotel, we could host research team from our member states. They could send students, they could send, of course, PhD students or small research team. And, of course, this is based on the shared things because, we cannot offer everything. So people have to come and offer manpower, offer new instrumentation, and so forth. So this is a concept of the research hotel which we are promoting now in the field of soft condensed matter and also in the field of science in extreme condition. And by extreme condition, because we are capable of reproducing the condition which we find in the center of the Earth in terms of pressure, in terms of temperature, but also we can deal with extreme conditions whenever we are talking with biological sample and macrobiological environment. And of course, this is aimed at addressing different issues, but in particular hydrogen storage materials and the stimulation of extreme conditions and so forth. And as we said, nowadays we have also to prove that with the science and the knowledge which we are generating, which are generating this large-scale infrastructure, has of any interest for the society. And of course, we are promoting the collaboration with industry. And not, of course, all the industries from our member states. I mean, people can come, they can ask the question, we can provide answer, we can help providing the answer. And so this is the structure which we are putting forward in terms of having a proprietary material characterization. So that's using X-ray and a neutron. That we can also complete research and development services. We provide, of course, scientific and technical expertise. We can also be considered as expert partners in EU framework programs. And we can also provide privileged access and to dedicated single point of contact. There is one single entry point for that. And of course, we provide confidentiality and IP ownership. So these are the new things which we are trying to propose. And of course, which can be integrated maybe in the overall strategy of Ceitec if you have a collaboration with industry, and this industry needs, access to the characterization of material in the condition which I described to you. Maybe this can be also a key point for collaboration. I'd like to finish here by saying we provide, this campus provide a wide range of spin-off in terms of training scientists and at a 3 years, 5 years contract. So this is like a little bit like EMBL. We have a postdoc program, we have a PhD programs, we hire very good students, they are being trained on the spot, and then they are moving and they are going to different places. In terms of training external PhD program, we evaluate that each year we receive about 1,000 PhD students who comes to the site. We propose also new technology for industry and innovative collaboration scheme, as I described to you, which are forming maybe the basis for further collaboration between Ceitec and the site. And of course, we participate in very proud, we are very proud of that, in the knowledge dissemination to national facilities in the institute of these 90 member states, which are our funding bodies. And with that, I'd like to thank you very much for your attention. Yes?
Thanks a lot. One quick question, as I promised. If any, if there is no quick question, I'd like to thank...
There is one question.
There is one? You have a strong voice?
Yeah, maybe I'll try. From the European Investment Bank. Question, because I know there was a potential project in Czech Republic. Just the last remark on national projects as a knowledge transfer. Do you ever see a future? Answer the question from all aspects, but from your perspective.
Well, it's difficult for me to intervene into your national policy, I know that for a while this has been considered. If you take a close look at the number of facilities which exist in Europe, if you take a look at the strength and the manpower and the resources which are needed to reach this level of competence, it took us about 22 years to be the best and remain the best. And we are dealing with 600 people, most talented people in terms of engineers and so forth. So this requires a tremendous investment. Whenever we talk about Europe, about the way we should use in a very efficient way the resources which are available, I'm not sure this is the best thing. I can tell you, in the field of crystal structure, people now are preparing their crystal. They send the crystal to us by express mail. Then we have robot. The robot puts the crystal in the X-ray beam, and the scientists can control the experiments from remote. More than 85% of the crystal structure of protein, which are deposited on the protein database, have been measured on synchrotron radiation facility. So things are changing. We are going into automation. And now we don't want people, we still want people to come to us, but we would like to offer to the synchrotron to go to our member states, by using these different progresses in technology. So this is my answer to you.
Thanks a lot. And now we have a coffee break, which will take 15 minutes, so we'll meet 10 minutes after 3 p.m.